We ready? Okay, so I'm going to tell you some stuff about how I use GFP in my lab. So just to review a couple points that Mohammed just told you about. So the green fluorescent protein originally was identified in jellyfish. And the key thing about GFP is that it fluoresces by itself. So if you put the gene for GFP into a cell, the cells make the protein folds up and then becomes a fluorescent molecule. So that's a really cool thing. We used to be able to use fluorescent molecules, we'd have to, a chemical, we'd have to tag things. But GFP is fluorescent on its own. And now we have a bunch of other colors of GFP, reds, blues, all sorts of different colors that we can use to visualize different things in the cells. And the way fluorescents work, as Mohammed pointed out, is that if you shine blue light on a fluorescent molecule, that photon, that blue photon is absorbed, it excites the fluorescent molecule, and then the fluorescent molecule then releases a photon. It loses a little energy in the process, so that photon is lower energy, different wavelength of light. For GFP, you shine blue light on it, it releases green photons. So it's good, you can look at a picture, if you have a microscope that's shining blue light on the sample, and then filtering out all the blue light and only showing green light, that microscope will only show you where the GFP is. It'll be black except for where GFP is. So that's one of the ways we can visualize GFP in different types of cells. So the relationship to your experiment and what I do in my lab, we're both using a plasmid to get GFP into cells and to have those cells express GFP. In your experiment, you're putting into bacterial cells that you'll grow on a plate, and you have another gene there, ampicillin resistance, which will select for the bacteria that have taken up the plasmid. So I'm using a very similar plasmid that has GFP, and I'm putting it into cells of the early developing nervous system, and they will express the GFP protein, and then those cells become fluorescent. And so these are some experiments that I did while I was at Caltech, and I was looking at how the brain develops in Xenopus tadpoles. So these are the South African clawed frogs. And so here you can see, here's the tadpole head, those are the eyes, here's the brain. We have GFP in there. Here's a close-up of it. If you blow that up and just look at GFP fluorescence, you can see these are neurons of the visual system. Here's the neuron cell bodies, these round things. It has the dendrites sticking up here. It has axons crossing over to the other side of the brain. So this is a tadpole that can totally swim around. It's totally fine. I actually trap it in Jello to look at it, and I can release it, and it's totally fine. So the GFP isn't hurting it, but it allows me to see the shapes of different neurons. So when I started my own lab at Pace University in New York, where I was just before I came to UNO, I switched from using frog embryos to using zebrafish embryos. So zebrafish are a small aquarium fish called danios. You see them in the aquarium shops all the time. They even have glowing danios, which are danios that have a fluorescent protein in them. And so I want to look at the brain development in these zebrafish in my lab at Pace. And so I'm going to show you a blow-up of here's the early developing embryo of the zebrafish. Here, again, I'm looking in the visual system, and those neurons are expressing GFP. And I'm going to show you a couple movies. I'm not sure if it's going to work here. Yeah, could you turn off the light? And so I'm going to open it up in... So this is a movie that's going to show you what it looks like if you were focusing down through the brain using a fluorescent microscope. So you can see as you focus down, you can see the cell bodies, and those are actually the dendrites of those neurons. And so you can see the shapes of neurons pretty clearly in that totally fine embryo that can swim around and do everything it's supposed to do. Here's a zoomed in version of that. And I'm going to loop it so it keeps focusing in and focusing in. And this just gives you a better view of those. And so you can see, again, the cell bodies here. These are their dendrites. They're putting branches out here. The axons from the eyes are forming synapses around here. So we get a lot of information about what the nervous system looks like as it's developing in these live zebrafish embryos. And so what GFP allows us to do is to look at this process in live embryos. And so we can put the GFP at this early stage before the neurons are extending all those crazy processes and stuff. And we can watch this process of how a neuron goes from an unspecialized cell to a specialized neuron that connects to all different parts of the brain. So we can literally see the brain wire itself up in a live embryo using GFP as a way to visualize that. That doesn't harm the embryos in any way, doesn't screw them up in any way, so you can see that process in live animals. And then what I do in my lab is do what we call a loss of function experiment. So we first look in a control situation and say, okay, if I put GFP in this group of neurons, this is what normally happens. They grow axons out this way and they grow dendrites out this way. This is the normal process that happens in development. And then we do a second experiment where at that first stage when we put the GFP in there, we also put something in there that screws up the function of a particular gene. And so we say, okay, this is the normal process. Now we're going to look at that process again and we're going to lose the function of a particular gene that we're interested in. We're going to say, does that gene, loss of that gene lead to this process no longer occurring as normal? And so it's a way to find out which genes are involved in this process. If you screw up the gene and it still happens normally, you can say, oh, that gene's probably not involved in that. If you screw up the gene and something happens, then you know that that gene may be involved. And so now I'm going to show you a couple videos where we did that. Oh, actually, let me show you. Sorry. Let me show you the control situation first. So here we're looking, again, in zebrafish embryos. This is the head, these are the eyes. And we're particularly interested in this case in the development of the olfactory bulb. So this is where the sense of smell first enters, and then the olfactory bulb is here. It sends axons back to other parts of the brain and brings that information about the sense of smell to other parts of the brain. And if we look at those axons in detail early in development, they come out and they cross over to either side and form connections in different parts of the brain. And that's kind of a 3D image of that. So we did this experiment where we first looked at what happened normally. This is what happens normally. Then we disrupted the function of a particular gene, one called DCC, and we said, does that normal process still occur? And so that's the videos I'm going to show you now. So first, in the control situation, here we're just, again, focusing down into the brain and seeing what we see. So if you focus down into the brain, again, you see that these axons and they're crossing over down at the bottom. If we focus in just looking at the green part, and again, I'll loop this movie so that it keeps going in. So again, if you focus down into that embryo, you see the axons coming out and they cross over and then send extensions out. So the olfactory bulb is up here. That's where the cell bodies are, sending out axons down here. And so that's what happens in a normal embryo. We haven't screwed up in any way. Now, what happens if we lose the function of this gene? So again, first the overall video that shows you where everything is. You can see there's axons coming down, but you don't really see those crossing over. And if we just look at the green, just look at the axons themselves, and I'll loop this again. You can see that we do see the axons coming down. They grow out the length. So we haven't stopped the growth of axons, but we seem to have stopped whatever it is that they're causing them to cross over. And this gene that we've disrupted in this case is a gene that's a receptor that senses signals and helps guide the axons go where they're supposed to go. So this is the receptor on the end of the axon. It senses that there's a hormone over here that says, okay, I'm supposed to grow to the other side of the brain. And so we found this gene that seems to be involved in mediating the crossing over the axons to the other side of the brain, and that's why we lose this process in the middle. So that's a quick, short example of one way that we can use GFP to visualize the development of the brain, and then we can use this loss of function approach to identify which processes are involved using that gene. And you're doing a similar process, not putting it into the brain, but put it into bacteria. So that's your first step. Next week, we'll get you doing brains. No, probably not next week, but soon. All right, that's it. Thanks so much.